Good day, everyone. This is Majida Pinto, and we are the Group 4. We are here to present our infographics about stool screening. But before we start, let us first define what is stool screening. So, stool screening involves the collection and analysis of fecal matter to diagnose the presence or absence of a medical condition. So, stool screening is used to detect the presence of disease-causing microorganisms such as pathogenic bacteria and aid in the diagnosis of a digestive system infections, gastrointestinal, and GI tract. It is a diagnostic test that determines whether diarrhea is caused by pathogenic bacteria, parasites, or viruses. A stool screening may be done in conjunction with other tests, such as a GI pathogens panel or an ova and parasite exam, to help establish a diagnosis of GI illnesses. When is it ordered? So your doctor could order a stool test if you show any of these symptoms. First, having diarrhea that lasts more than a few days. Second, a poop that contains blood or mucus. Third, stomach pain or cramping. Fourth, nausea. Fifth, throwing up. And lastly, having fever. So if you show any of these symptoms, then it is required that you do a stool test. How is it examined? So first is by saline specimen preparation. In saline specimen preparation, we obtain a microscope slide and the stool specimen, take a small amount of the specimen and place it on a microscope slide. If the stool specimen is still somewhat solid, we add a drop or two of saline to the specimen and we mix it. Saline specimen preparation is employed primarily to demonstrate worm eggs, larvae, protozoan, trophozoites, and cysts. Second is by iodine specimen preparation. An iodine wet mounts were made by combining a little amount of stool sample with a drop of Lugol's iodine on a glass slide, uncovering it with a cover slip. Iodine is a non-specific, fast contrast dye that is used to distinguish parasite cysts from host white blood cells in direct wet mounts of feces. And lastly is by concentration method. Concentration procedures separate parasites from fecal debris and increase the chances of detecting parasitic organism. Now the second reporter will be Siti Noraisa Pascan.
This is Siti Noraisa Pascan.
Worldwide parasitic diseases is increasing, therefore timely screening and optimal quality of the sample stool are important. Thus, the correct collection method of stool is needed. To ensure there is no contamination, the sample stool must not get in contact with urine, any toilet impurities, for this may affect your laboratory result. With that, a garbage bag or a biodegradable flash paper and the likes can be used in catching your stool. Collect the sample just the size of walnuts and put it in your prepared container labeled with your name, birth date, and the date. Clean the surface of the specimen bottle with tissue paper and not alcohol. Seal the specimen bottle properly to avoid any leakage. Dispose your garbage and clean your hands thoroughly with soap and water afterwards. Return the sample to your doctor's office as soon as you can. Tell your doctor about any medicines you're taking. What does the result mean? If this is negative, which means there is no bacteria, parasites, and other biomarkers found on your stool. If it is positive, bacteria, parasites, or other biomarkers have been found on your stool. With that, you must connect with your doctor for further investigation while following a healthy lifestyle.